Comprehensively characterizing the tumor microenvironment is essential in immune-oncology research. This assay is the first to use RNA-based health expression models to measure immune cells in solid tumor tissue.
This assay enables researchers to gain highly sensitive and specific measurements of the immune contexture in FFPE solid tumor tissues and to combine these results into a multidimensional biomarker.
All cancer treatments, not just immunotherapies, elicit an immune response at the site of the solid tumor. Measuring this immune response is important for understanding disease progression and therapy responses.
This protocol combines traditional RNA library preparation techniques with targeted capture, while time-consuming following the wash steps and monitoring the quality control checkpoints as suggested is very important for success.
For highly degraded RNA from formalin-fixed paraffin-embedded samples, assemble the first strand synthesis reaction on ice according to the table. Thoroughly mix the reactions by pipetting up and down several times before briefly spinning down the samples in a microcentrifuge. At the end of the centrifugation, immediately place the samples into a preheated thermal cycler following program number 3 from the table. For high-quality intact RNA, assemble the first strand synthesis reaction on ice in a nuclease-free PCR tube as outlined in the tables. To begin this procedure, mix 200 nanograms of the barcoded